Hi, this is Denis Wirtz. I'm a faculty at Johns Hopkins University, and more important here, I'm a member of the Johns Hopkins Physical Science and Oncology Center. Um, Along with my good colleagues, Sharon Geher, Kostas Konstantopoulos, and Sean Sun, we started this center some 11, 12 years ago. And the vision of the center was really to uncover the physical underpinnings of cancer metastasis. So truly addressing one of the most important and unmet need in oncology, that is metastasis, which is responsible for 90% of cancer deaths. From the get-go, we ensured relevance in cancer by bringing along oncologists and pathologists, including Ken Pienta, Greg Semenza, and Greg Longmore. So work in the center really seeded into a new field of basic science and translation, leading to, as you'll see in a moment, new treatments and improved prognostication tools of use for oncologists and pathologists. One of our main contribution was really, first and foremost, delineate what this metastatic cascade may look like. And so how migration, in particular migration in 3 dimensions, could really uncover potentially new molecular pathway for cancer cell migration and eventually translation for anti-metastatics. We were one of the first groups to really envision how nuclear and cell volume and how water, indeed water coming in and out through channels and via ion pumps of the cells could modulate mechanosensation, the ability of cells to sense stiff and soft microenvironments, migration, differentiation, and polarization. These brought about completely new predictive theoretical model and uncovered new molecular targets, including ion channels and receptors. Along the way, we developed new technologies that facilitated these discoveries, including highly tunable hydrogels, high throughput single cell phenotyping technologies, microchannels for studies of confined migration, and machine learning-based automatic measurement of cell volume in live cells. Translation was and has been always a focus of the center, including patents, spin-off companies, and most importantly, new therapeutics and prognostication tools for cancer patients. In particular, I'm proud to say that the center is now developing first of its kind biospecific antibodies targeting specifically metastasis. The work was accomplished with brilliant PhD students and postdocs who now have moved on and graduated from the center. I'd like to highlight Stephanie Fraley at UCSD, Daniel Gilkes, who's now a colleague at Johns Hopkins, Yizheng Li, Panos Mistrolitis, and Jude Philippe, also a colleague at Johns Hopkins. The center fostered really early on the career of young faculty. Sharon, who's now a member of National Academy of Medicine, Karen Eisiger at Penn, and Pei-Shun Wu, another colleague at Johns Hopkins. We've had myriad of interactions across the network, leading to landmark publications in mechanobiology. And finally, I'd like to acknowledge really visionary Anna Barker and Jerry Lee, who believed not only in us, but believed in the idea that engineers, physicists, chemists, computer scientists could bring among new ideas, new vision to oncology to bring new treatment to them. I'd like to also acknowledge Larry Nagaha and Naz Zair, who helped really chaperone a wonderful network of interactions that really literally change our lives. And finally, I'd like to really thank deeply Naz Zair, who from the very beginning not only helped create this network, but also brought it to make it truly relevant in oncology. So again, thank you. And I would say happy birthday.